Welcome to class number 9, session number 3. In this session, we will start seeing examples of sensors that are based on polymeric nanostructures. Conducting polymer sensors are based on the variation of their capacity, optical properties, redox properties, etc., etc. The sensitivity of conducting polymer-based sensor is primarily determined by the difference in a specific property before and after exposure to a target molecule. The specific surface area of conducting polymers plays a key role in determining the sensitivity of the sensor. Conducting polymer nanostructures such as nanotubes, nanowires, and nanoparticles offer new opportunities as sensing platforms for biological and environmental applications. Of these opportunities, biosensors are of great importance. As mentioned in the previous classes, a biosensor couples an immobilized biospecific recognition or catalytic component to the surface of a transducer, which then converts the biological recognition event into a measurable electrical signal, pinpointing the presence of the target analyte. In the presented scheme, you can see the application of different conducting polymer nanostructures in biosensing. In A, you can see a biosensing based on different conducting polymer nanostructures, and then you can see biosensing that are based on single conducting polymer nanowire. The high contact area between the analytes and the nanostructures result in enhanced and amplified sensitivity of the biosensing. Here we will present on this screen the new developments in the field of biosensors that are based on nanostructure polymer. Researchers have introduced a conducting polymer nanojunction sensor for glucose for potentially serve as useful tool for the in vivo detection, since it's very specific and responses quickly towards glucose. The sensor consists of an array of polyaniline nanojunctions. Each nanojunction is formed by electropolymerization of polyaniline in the presence of polyacrylic acid, namely PAA, to bridge two nanoelectrodes separated within a nanometer scale gap that range between 20 to 60 nanometer. Polyacrylic acid allows to maintain significant polyaniline conductivity near the neutral pH. In this case, the glucose oxidase is immobilized into the polyaniline polyacrylic acid to provide specific detection of the glucose. Upon exposure to glucose, the glucose oxidase catalyzes the oxidation of the glucose and becomes reduced. The reduced form of the glucose oxidase is regenerated throughout reoxidization by oxygen in the solution, which produces basically the hydrogen peroxide. Hydrogen peroxide then oxidizes the polyaniline and triggers an increase in the polyaniline conductivity because of the sensitive dependence of the polyaniline conductivity on its redox state. The results of this process are described in the present slide. In panel A, you can see the structure of the polymer nanojunction sensor, and in panel B, you can see same image of the nanostructured polymer films deposited on gold pads with 20 to 60 nanometer gaps. In panel C, you can see the time course of drain current at the nanojunction upon successive additions of 40 millimolar glucose, and in panel D, you can see the corresponding calibration plot of drain current changes versus glucose concentration. The second example of a biosensor is the gene sensor that is presented on the screen. So far, a group of researchers have constructed a transducer that is based on copolymer of P dot and carboxylic group functionalized p-dot single nanowire in between gold electrodes, followed by covalent attachment of amino-modified prop of short DNA that is specific to breast and ovarian cancer cells. The results shows that these short DNA molecules are detected at femtomolar concentration, and the negative controls are clearly discriminated by the sensor. The ex situ measurements are performed by using two-terminal device setup, and the changes in the interface of the nanowire associated with the association or disassociation of the oligonucleotides or short DNAs were measured as change in resistance. The left figure shows a schematic representation of the two-terminal copolymer nanowire sensor. In panel A of this figure, you can see a single copolymer nanowire that is grown between gold electrodes. In panel number 2, you can see covalent attachment of the prop short DNA on the surface of the carboxyl-modified copolymer nanowire. In panel D, you can see the hybridization of the targeted short DNA with prop-modified copolymer nanowire. And in panel number 4, you can see the measurement of the change in resistance across the polymer nanowire by applying a sweep voltage between the gold and the electrodes. Another important example of solution-based sensor is based on optical solution sensors. Suspended core microstructured optical fibers with the photo-induced electron transfer, or which we abbreviate usually PET, effect are combined to demonstrate a new type of fluorescent optical fiber deep sensing platform for small volume ion detection. A sensor design based on a simple model PET fluoroiono 4 and a small core microstructured optical fiber capable of detecting sodium ions is demonstrated on the screen. The molecules loaded in the holes of the device are excited by the portion of the guided mode of the 473 nanometer pump light located within the holes. A fraction of the excited fluorescent photons emitted are captured within the core and guided to the fiber ends, where forward and backward propagating fluorescent signals can be detected. In the experiments reported here, one can detect backward propagating fluorescent signals to utilize the left-hand side fiber end as a deep sensor. As expected, the right figure shows that the intensity of the excitation and emission spectra both increase with the increasing concentration of the sodium ions. No significant shift in the wavelength of the excitation or emission spectra occur as concentration of the sodium ions increased, showing the targeted molecules does not exhibit photo-induced charge transfer and therefore is operating throughout photo-induced electron transfer mechanism, or the so-called PET mechanism. Another example of the optical sensors that can be utilized in solution phase, or liquid phase basically, are presented here and based on complexes of conjugated polyelectrolytes and in organic semiconductor nanoparticles. Let's now take a look on the bottom figure capture. In this case, the silicon nanoparticles are prepared by pulsed laser ablation of silicon wafer in aqueous solution containing cationic surfactant of CTAP. In this case, a layer of the conjugated polyelectrolyte is assembled on the silicon nanoparticles, and then 20 millimolar surfactant of CTAP is added. Complexation of the polyelectrolyte with the charged silicon nanoparticles is formed by gradually adding the silicon nanoparticle stock solution into the solution of the conjugated polyelectrolyte. Let's now take a look on the figure caption on the top figure. In this case, you can see the influence of increasing the concentration of the cytochrome C on the fluorescence spectra of dual fluorescent nanocomposite of the silicon nanoparticles and the conjugated polyelectrolytes. In this case, the arrow indicates the direction of the change in emission intensity with increasing the concentration of the cytochrome C. Fluorescence emission of the hybrid silicon conjugated polymer nanocomposites is steeply quenched as cytochrome C, and the minimum detection concentration for the redox active protein is found to be 50 nanomolar. The sensitivity of this tool is realized by ultra-fast photo-induced electron transfer between the electron-deficient protein and the conjugated polyelectrolyte binding on the silicon nanoparticle surfaces. Please note that this is because that the conjugated polyelectrolyte has a large number of ionic groups and aromatic rings, which enable strong adsorption. Mechanical-based sensors are an important part of the liquid-based sensor industry. In the present research, the pH nanosensor is based on the real-time determination of swelling or shrinking of responsive ultra-thin polymer brushes by an inexpensive, simple, efficient, and highly sensitive technique in which gold nanoparticle enhanced transmission surface plasmon resonance spectroscopy. The approach is illustrated by the observed 50 nanometer shift of the SPR, namely the surface plasmon resonance absorption, maximum by the swelling of poly-2-vinyl pyridine polymer brushes caused by changing the pH from 5 to 2. Changes in pH generates more charges in the interior of the polymer brushes, which are then forced by electrostatic repulsions to stretch out and hence to swell to a thickness several times greater than that for the uncharged polymer. Please note that in the top figure, you can see schematics of the reversible pH change-induced swelling of the gold nanoparticle coated with poly-2-vinyl pyridine polymer brushes. And on the bottom figure, you can see SPR spectra of gold nanoislands containing adsorbing polymer brushes and gold nanoparticles at pH that range between 2 and 5. We reach now the end of class number 9, session number 3. Thank you.